Thank you, everybody, for joining us today. I'm very excited to be able to introduce this topic of molecular epi of viral pathogens. I'm going to try and take a almost like a 30,000 foot view of this topic and try and give you guys a little bit broader skills and knowledge to interpret molecular epi and to understand how where we are now and where we're hoping to go. Uh, I'm speaking this week. My name is Justin Ball. There's my email address. Feel free to reach out if you have any questions that we don't get to today, or if you'd like to have a little bit more information. Next week, we have Nate Gruba who's going to be speaking, and that's very exciting as well. He's an expert in molecular epi of vector-borne diseases, and he's going to try and give us a different area. My expertise is on respiratory viruses. Let's see. There we go. I'll introduce myself a little bit more in the next slide. But today, just to acknowledge this challenge that we all have, not just in viral molecular epidemiology, but epidemiology in general, and the use of genomic data to try and better understand outbreaks and outbreak dynamics. It is a big challenge. We do have to make sure that we're all speaking the same language between these different domains, from the laboratory technician all the way up to uh, the epidemiology branches and leadership. So integrating the knowledge across these different domains requires a really broad range of expertise. And we have to bring in the technicians, the bioinformatic computational biology support, enhancing computational infrastructure, creating an integrated data management, and then method development and validation. This is an enormous challenge, and as we're moving towards real-time molecular epidemiology, it's important to acknowledge that we're not quite there yet. That a lot of these areas need to be developed quite extensively before we really get to the point that we want to be, where we have real-time molecular epi. So this is me. I am a professor in the Department of Infectious Diseases, the Department of Epidemiology and Biostats, and the Institute of Bioinformatics. I lead the Applied Molecular Epidemiology Research Group. We are a group of collaborators and trainees that are trying to understand the transmission of RNA viruses across biological scales. We focus on influenza, coronavirus. We've dabbled in dengue and HIV, and then we also do a lot of work on animal pathogens and emerging pathogens as well. All trying to use genomic data to better understand the reason why we're seeing the diversity of viruses that we do see, and hopefully to understand how those evolutionary processes are determining the outbreaks and the different character evolution, like vaccine escape. So I'm going to go all the way back to the beginning here. I'm going to start with what is epidemiology. So it comes from the Greek term epi, meaning upon, and demos, people, and logos. So it is literally the study of what is upon people. Uh, and this is where we're starting from. We're not inventing something new. We are trying to understand those five W's. And I think it's important to remember that ultimately we are trying to answer these questions in epidemiology, and we are using genomic data to try and do that. So we want to know, what is that health issue concern, what people, or who in the population is being impacted, where are these epidemics occurring, when are they happening, and try and understand those causes, risk factors, and modes of transmission in the whys and how. Molecular epidemiology is a branch of epidemiology uh, and biomedical sciences that come together by merging molecular biology into epidemiological studies. So essentially, we are still answering those same questions, and our aim is to identify those infectious agents, unravel the source, identify their reservoirs, trace their circulation patterns, and the transmission patterns using genomic data. The slide. Okay, I'm seeing your comment now. Hmm. Yes, I can do that. I'm gonna slideshow. This is the problem of using three screens. Let's see. Is this a little better?
Awesome. That looks good. Thank you.
Okay, thank you. Sorry, and the other bonus of using three screens is that I've got all this uh, real estate to view things, but Zoom disappears when when I'm presenting. Okay, so I'm glad this is better. So the aim is again to identify the sources of infectious agents, what they are, the source, identifying reservoirs, maybe tracing circulating patterns, and the transmission patterns, maybe identifying what part or what's the structure of the population and the transmission. All these things that are determining the pattern of outbreaks that we see and the patterns of genetic diversity that we get from the sequences. And our goal is to try and get to this real-time genomic epidemiology. So this is just a basic workflow. We have samples that are collected often through clinical surveillance. These are sequenced. We need to create a multiple sequence alignment. This is not an alignment free. So the first step, once we've done the QC on the sequencing, we have to identify, we have to build a database of aligned sequences. And then we can do a comparative analysis. We are comparing those sequences against each other and often building a phylogenetic tree. Once we have that phylogenetic tree, we can start investigating some of those epidemiological processes that might determine the shape of the tree, as well as putting this information into models of to estimate that epidemiological transmission within the community and investigate some of those processes or population structure that might be shaping that outbreak. One thing that we've done very well with molecular epi is looking at the spatial epidemiology. Um, and we often talk about phylogenetics and phylogenetic spatial reconstructions. These work very well at the global scale, and I think we've been able to effectively understand the pace of global transmission as well as tracing variants as they move between these different areas. And we've also done a good job of investigating the role of human movement patterns and uh, from the individual level as well as mobility patterns like the amount of flights going between countries and incorporating that into our molecular epi studies. But as we get down to within a specific population, it is much harder to effectively extract that information. So when we're looking at molecular epi within a particular community, it's really hard to effectively understand and use genomic data. So just for your reference, using molecular approaches to understand transmission, in particular of uh, viral outbreaks within a community, is actually very difficult right now, and that is an area that our group has been actively investigating, as well as many others. And I think that's sort of the forefront right now is developing those methods to try and understand how we can use genomic data to answer those questions. But we have done a very good job of tracing them across that global scale. So some of the specific topics that I'm going to try to cover today is interpreting phylogenetic trees and an introduction to phylodynamics. Uh, we'll touch a little bit on that spatial epidemiology further and emerging and epidemic diseases. Uh, oh, I apologize. This is an incorrect slide. This is not all the things I'm going to cover today. I'm mostly going to talk about the background and interpreting phylogenetic trees, and then just introduce some of the spatial epidemiology across scales for this talk. So, first thing, what is a virus? This is the definition that I use for a virus. It is an infectious obligate intracellular parasite comprising genetic material surrounded by a protein coat and or a membrane. The reason why I use this definition is that this saying that it's an obligate intracellular parasite, that means that we recognize that a virus and a virion are two different things. That the virus is an organism with two phases. So a virion, and then we have the virion, and then we have the infected cell. Okay, and the virion, in order for it to survive and spread, it has to get into the infected cell. The genetics of that virion is restricted by the fact that it has to be able to replicate within this cell. If you have a salad for lunch, you will be exposed to lettuce viruses. Those lettuce viruses cannot replicate in your cells. So there's no risk to you. You've not been infected with them. It's restricted by that host genetics. But the other thing is when we look at these things as this organism with two phases, means that when I am studying the genetics of the virion and when we are studying the genetics of the virion, we are actually learning about the infected host. So when we make inferences on geographic movement, we are learning about movement patterns of human populations. If we're doing this for high path avian influenza and in wild birds, we're investigating the migratory patterns and the behavior of those wild birds by using this genetic material. Now, infectious diseases have played a major role in human evolution and societal development, and they're not new, of course, and certainly SARS-CoV-2 was not the first pandemic that we had been faced with, but they played a very large role in human development, especially smallpox, which I think the first recognized outbreak is 430 BC, having killed more than 30,000 people in Athens, Greece. 1519, we still have this massive outbreak of smallpox, killing between 5 and 8 million people, native population uh, in the Americas. 1918, we have the Great Flu pandemic, estimated to have killed between 30 million and 50 million people. 1952, we have another polio. We have a polio peak at this time with 60,000 children infected, and of course, impacts on mobility and people needing to use a iron lung for very long periods. 1984, we recognized HIV and a cause of AIDS and the global epidemic that was triggered by this virus. 2003, we have severe acute respiratory syndrome that started in China and having spread to large parts of the world. And then 2009 uh, is when we have the H1N1 pandemic. Everything before this period did not rely heavily on genomic sequencing, right? We've been able to do retrospective studies and investigate how the genomic data has been associated with these different outbreaks. But really, in 2009, this is the first real outbreak where uh, molecular epidemiology becomes the primary tool for learning about that outbreak in real time in the early part of the epidemic. Uh, we've had a number of epidemics in what we can consider the genomic age. Of course, we've had a number of chikungunya outbreaks. We've had Ebola. We've had the reemergence of Zika virus. And of course, we've had an outbreak of MERS and measles. We've also had a number of outbreaks of avian influenza, both high path H5 and H7. We've had human infections of H10 and H9, these different subtypes of influenza, and we're investigating all of those primarily with molecular epi. When it spreads through populations more broadly, then we're able to investigate these outbreaks with traditional boots on the ground epi. But the early parts of these epidemics, of these emerging epidemics, is often driven. The knowledge is often driven by genomic sequencing and comparative genomics. So that swine flu epidemic in 2009, this is one that I was directly involved in my first outbreak that I had worked on, or first pandemic I had worked on. The timeline of this is that April 15th and 17th, the novel swine origin influenza was identified in two unlicked patients in the United States. That same strain was later identified in Mexico and Canada, and it spread across the globe in a matter of weeks. The global network of scientists were made aware of the new threat on April 23rd, 24th, and through monumental efforts from CDC, those first sequences of that H1N1 virus were made available to the community in April 25th. The outbreak was investigated, data was shared very quickly, very easily, and the group that I was working with, we were based in Hong Kong. We had our partners in the UK and partners in the US. We analyzed all of our data through an open access website, providing all that information to the global community. And it became a place where scientists came to discuss the outbreak and the analyses in near real time. We would work for about 8 hours. We would hand it off to the group in the UK that they would start leading their analyses and commenting on the work that we had done in the previous 8 hours. And they would update their analyses, and then the US team would sort of take the lead in that following 8 hours. So it was it was very open in how we were analyzing that data. And we were able to investigate the source of the epidemic, looking at this model coming from wild birds, the interaction between wild birds, domestic animals, and spillover into humans, and trying to recreate the genetics and the origins of this pandemic virus. We were able to determine that we had viruses that were circulating in wild birds that managed to get into European swine. We had a collection of viruses that were in North America that had ancestry with the 1918 pandemic. And then we had human seasonal influenza viruses that had spilled over into swine as well. Subsequently, we had avian spillover into this North American population, and a couple genes from the European swine-like population had ended up into the swine population, but wasn't detected until it actually emerged within humans. So we were able to use genetic data to trace how this virus had emerged and how it had moved between these populations, and adding more detail to this particular model of pandemic influenza emergence that wasn't available before. So if we think about these different eras, we have the pre-germ theory era, which is an era of plagues, and we have development of early epidemiology. This leads up to this period of discovery when we start understanding about bacteria and viruses. We have the post-germ theory era, which is really where modern epidemiology sort of takes root and is developed. And now we're moving into this new era, which I just tentatively named the modern era. Maybe we should just call it the molecular epi era. But this is an era that's driven by massive amounts of data, analyses that are open source. We're taking advantage of these molecular approaches, and the pace of discovery is extremely fast. If we think about SARS-CoV-2, at one point I just was curious how many papers had come out that week, and we were looking at more than between 4,000 and 6,000 papers coming out every week. So the pace of discovery is very fast. A lot of that was built on genetics. Of course, there was other experimental work in there as well, but the pace of the development of this field is extremely fast. Oh, that's a so again, since we have this virion that we're studying the genetics here, and we're learning about the host as well. But we have comparative genomics tells us something about these different systems, but it is very complicated. Ultimately, we are trying to understand how these viruses move between biological scales. So when we have viruses in an individual, that is the biomedical scale. We might consider this the genetics here to look at the emergence of vaccine escape within an individual, maybe to understand how diversity is being generated or how error prone the virus is within that individual. It might be escape from antivirals, or it can also be looking at how the virus diversifies in different organs as well as a number of other approaches. But really, when we're looking using genetics within the individual, we're at the biomedical scale. Linking the individual to the population scale or the epidemiological scale is very difficult. But we are using this data, especially during SARS-CoV-2, to investigate how the viruses move through a community and through a population, whether there is age structure within that spread, or gender structure, whether it may be determined through human behavior like sexual contact, as well as maybe other factors that are determining that. When we start considering how viruses move between populations, we're moving into the ecological scale. And if we're looking at pandemic emergence, we're moving between these different host species in different and different populations again. So that's also an ecological scale. Evolutionary processes um, determine the amount of genetic variation that we see at a given time point. When we are doing comparative genetic analyses, we're looking at that genetic variation. So I just want to remind you that there are four basic mechanisms for evolution to use, and these are mutation, migration, genetic drift, and natural selection. We've probably heard natural selection. If we have a huge amount of diversity of viruses, and one of those viruses is more fit, however way we want to define fitness. If one of those viruses are more fit, that will become the dominant virus. What that means is natural selection is going to actually actively reduce the amount of genetic variation that occurs. And we'll see that a period of high genetic diversity, natural selection, one variant survives, and we have a single dominant strain that comes forward. When we sequence a population of viruses, we're observing that genetic diversity that has resulted from those evolutionary processes. And it is just a single snapshot in time. We can collect a whole bunch of viruses over time and build a time structured phylogenetic tree, but taking uh, all those snapshots in time just give us an idea of how some of those how the genetic variation might change over time. Okay, but it is just a series of snapshots. To view these things, we often build phylogenetic trees. A phylogenetic tree is an evolutionary hypothesis. It is not just a drawing. Okay, it is an evolutionary hypothesis that shows the history and relationships through a process of common descent of the observed genetic diversity. Okay, so a phylogenetic tree, it is an important evolutionary hypothesis. The From the first drawings of the phylogenetic tree is from Charles Darwin. This was written in one of his notebooks. And Darwin and Wallace formulated the theory of evolution by common descent. 1859 is when we have the origin of species, and then we have a modern synthesis. Now, I mentioned these different biological scales. I'm going to try and lead you to them actually being linked. So, ecology proposes mechanisms for genetic variation between populations. Epidemiology studies the patterns and causes and effects of disease on populations. And an ecological study is an epidemiological study in which the units of analysis is a population rather than an individual. Okay, so we're in an epi study, we're looking at transmission between individuals. When we say we're interested in transmission between populations, we can actually consider them equivalent, except for that unit of study that's being observed. So we try to make inferences on the ecology, the epidemiology, and the evolution from our phylogenetic tree. So here is a toy tree. I'm going to just walk you through how we actually read a tree and some of the interpretations that are made. So this is our toy tree. Our toy tree has dimensions. Okay, branches are different lengths. Well, they tell us something. First thing that it tells us is that I want you to know is that vertical dimension is non-interpretable. There are efforts to add data in there, but in a basic phylogenetic tree, that vertical dimension does not have meaning. So you really just want to focus on the horizontal dimension in this orientation. And that horizontal dimension tells us how much genetic change has occurred on a branch, which can also be equated to time, units of time, or just like the longer a branch is, the more genetic variation has occurred on that branch. That is equivalent to a greater amount of time that has occurred between its common ancestors. That scale bar, and whenever you make a phylogenetic tree, always include a scale bar. Okay, the scale bar shows the length, in this case, equivalent to nucleotide substitutions per site. Okay, so it's saying 0.5 nucleotide substitutions per site. It could be units of time. They could be epi-week, they could be months, they could be years, or nucleotide substitutions. Always include your scale bar when you produce a phylogenetic tree. The structure of the tree. Well, I'm calling this a tree. Trees have leaves. So we have the tip, the taxon, the virus, or the leaf. This is actually our sequenced viruses. This is where they occur on the tree. We often know things about that virus, which help us interpret the tree, and they can actually be modeled directly onto the tree as well, if that's appropriate. So we might know the date, the location of isolation, the actual host it came from, or maybe clinical features. Including this information makes these analyses way more powerful. And if you want to use a phylogenetic analysis within a community, you're going to have to include more metadata that might be associated with that sequence. For example, maybe it's the zip code, the exact date of isolation, maybe the age of the individual, or other features of that population, and clinical features. This is also where we need to make sure that we have IRB approvals to share and use those data sets. We have internal nodes, just like trees, they have internal nodes. Internal nodes are where two branches come together. Okay. Here, these are the putative common ancestors of the two observed leaves. And then if we follow a tree from a leaf all the way back through all its nodes that come together and all the branches that keep going, they always come all the way down to a root. The root in a phylogenetic tree, when you root a tree, it provides an order of these branching events. If you use a program like Beast, it estimates the best place for that root based on the data that you give it. If you're not using something like Beast where you're going to try and model where that root fits, you often want to use an organism that's a little bit distant from your study population, your group. So if you're looking at high path avian influenza within a outbreak in, say, in Athens, Georgia, which is where I am, you would take all those sequences from Athens, Georgia, and you might include a sister virus, or one that shares a common ancestor, like the H5N1 virus in 1996 that was isolated in China, which is the common ancestor for all these other ones. That will provide a order of which these branching events are going to occur. So we do want to have a root. It helps us to interpret these trees. Some terminology. If we're interested in the green group and we say, oh, all these green groups, they share a common ancestor, we would say that that is a monophyletic clade. If that green group includes some of these blue groups, we would say the green organisms are paraphyletic, and they descend from a common ancestor, but they include other blue individuals. So just reading a tree, maybe we're interested in this trait, viruses from cold environments, viruses from hot environments, and we would say, well, all the hot viruses grouped together in this circle, and all the cold viruses grouped together in this second circle. But then we build a third circle that includes all the traits that we see within the tree. Maybe we're interested in geography. So we would say, oh, Antarctica, the Arctic, and South America, these places with cold environments are all, or in this case, these ones grouped together, and then these ones in Asia, North America, and Africa grouped together, and then we build a larger circle around all of them. Okay, so we would say this blue group shares common traits, and this yellow group shares another set of traits. Maybe we want to try and look at an outbreak. So if we just go through the steps here, we have hospital 1 and hospital 2. Let's imagine we're tracing a antibiotic resistance between these two hospitals. And we say, okay, well, here's our first two individuals. They clearly share a common ancestor. But then we have to include these two other individuals sharing another common ancestor. So we're expanding our circle. And then we build this other trait here where all these individuals share that common ancestor. So we would say that hospital 1 and hospital 2, the outbreak has likely moved between these populations, and they are mixing right now. The shape of the phylogenetic tree itself is really structured by host ecology and epidemiology. So human influenza has something that's known as this classical ladder-like phylogeny, where many tips on the tree are generated during every epidemic, where all individuals are, or many individuals are infected, but most of those do not actually persist in the population. So they are removed likely through a combination of human migration uh, and human immune selection. So we have actually at a given time point, if we just take a little line where my laser pointer is, we actually have very little diversity at one time point. And moving through the whole tree, we don't actually get that much diversity of influenza, but all those ancestral variants are constantly replaced by new ones. These are all trees that I built using H3 viruses. So they're all the same subtype. If we look at H3 in avian influenza, in wild birds, we get these really long branches that are structured by geography, continental regions. And this top one is a historically North American lineage. The bottom one, or the middle one here, is a Eurasian lineage. And then we've had an introduction into North America. So now we have two lineages circulating in North America, one that shares common ancestry with these ones in Eurasia. If we look at H3 viruses in poultry, we often have long branches that are connected by an explosion of tips. Almost looks like a palm tree, each of these individual branches. So we have uh, a exponential growth of an outbreak, often spreading within a farm or between farms in a very localized area. But we don't have viruses persisting over very long periods, but we do have long branches without any data, indicating that they're coming from someplace. Uh, the the shape of the tree, as I mentioned, it's also part of this. It is also determined by some of that epidemiological process. Uh, what I mean by that epidemiological process is that we have transmission through a community, and if we build a transmission tree, we could see how a virus, if we were able to sample all individuals and trace them through all time, we would see that a virus goes from one individual, it infects this second individual next, and then this individual here. This individual goes on to infect another individual. This individual comes down here and affects someplace else. This one continues on, and we can trace that epidemic all through time. Uh, if we understand how that within host evolutionary process takes place is that we have amount of genetic variation that we observe within this individual. We have transmission. We can have new mutations occurring, and then we have transmission again. Transmission or mutations occur along these branches as it replicates within an individual, and then sometimes those mutations are transmitted to new individuals. These blue cases are the ones that we observe through our sampling. They're the ones that showed up at the clinic. They are the individuals that were part of an epidemiological investigation. So these are the ones that we end up sequencing. We can imagine that if we look at these two individuals, they have a common ancestor somewhere back here in time. This individual has a common ancestor with all three of these individuals, and then all of these individuals would have a common ancestor somewhere back in here. So we don't actually observe those common ancestors, but we can start estimating the transmission tree from the actual sampled sequence cases. So just back to our toy tree, when we have that metadata and we start understanding these traits, maybe we want to look at, say, green and purple individuals. Is there a structure based on the set of colors that I have here? Can we start identifying a maybe a trait? Maybe this is the index hospital. That the red was the index hospital. Maybe it's a party, a costume party, as it moves to these individual cases in the population. So we ultimately we want to be able to link this tree to these to the actual transmission and linkages between these populations, and that remains a really big challenge. Phylogenetic is this field where, because viruses replicate so quickly, they transmit so quickly, and they accumulate mutations so quickly that their evolution, their ecology, and their epidemiology occur on the same time scale. Because of this, we need to study them together. Uh, and this field of phylogenetic was introduced in 2004, so it's still a very new field, uh, and it has grown really rapidly. Almost um, new methods were being developed very quickly, new approaches, and new tree building methodologies, and trying to incorporate all the information. Uh, what we saw during SARS-CoV-2 was that we suddenly had more data than our existing methods could handle, and we had to develop some new approaches. But phylogenetic, when you do hear this term, it is the goal of phylogenetic is to start understanding how those ecological and epidemiological processes interact to determine the shape of the tree and the patterns of diversity that we see. Couple points here. All observations, these assumptions, we assume that all of our observations are related by common descent, and that we can describe evolution in this bifurcating tree. And then we interpret it by saying that when we have really small genetic distances, that means that these groups are closely related and therefore connected by a common source. Okay, so that's just to keep in mind. We do have situations where evolution is not bifurcating, especially when we have things like recombination or reassortment. That confounds this approach. Uh, and when we have a group of viruses and we have something that's really unrelated, and we have a bad data set, they may actually not be linked by common descent, right? So that goes into actually how we've collected our data. We assume that it's linked by common descent, and this approach will always produce a tree, okay? But that might be wrong if your population of viruses are not. This will actually become a really big issue in wastewater surveillance, and that we are just blindly collecting all the sequence data and trying to interpret them, where we may have individual sequence fragments that are not linked by common descent, but we don't have a good way of removing them from our analysis. So it's just something for you to consider as time goes on. So far, really, all these approaches have been very um, qualitative, right? We look at the tree, we can build a tree, and then we can look at the tree and say, well, these individuals share a common ancestor. This is our trait of interest or our population of interest, and we can start making some inferences. But really, the major challenge that we have right now is integrating all these different types of data and developing it into a statistical phylogenetic approach. So if we were going to build a pipeline, we want to take sequences directly from that sampled population, clinical samples, uh, build a phylogenetic tree, infer an epidemic network, and then start doing some statistical hypothesis testing. Is that network actually a good representation of the test? Have we actually have we actually determined what is important from this network, and then trying to model this new information onto that tree? This is a tough thing to do. We've done it very well with geographic location. We assume that the patterns of genetic diversity follow the geographic history, and we assume that the signal of geographic spread is recorded in those genomes. These so far seem like pretty good assumptions, and when we've done the phylogeographic reconstruction, it's worked out really well. But like I said earlier, trying to get into that local scale is really, really challenging. And we don't know whether our sampling is even good enough or how we might model these other traits. So this is a really big challenge. And then on top of that, data sets are getting bigger and bigger and bigger. Uh, it's not impossible, but these are all the steps that we actually need to start doing to really be able to say an outbreak started at a school, is transmitting primarily among school age children, and they're the characteristics that's in this geographic region, all these other things, in order for the epidemiologist to say, given this information, this is the best control measure, right? So we're trying to get all that information out there, and we are making really good strides towards achieving that. I just want to highlight the last couple slides here, and I know I've gone through a lot of material very quickly. Hopefully, we can chat a little bit more about it towards the end. But uh, just one of the approaches that we've taken to try to understand the viral transmission at those regional and local scales for SARS-CoV-2 is we actually started developing some new metrics and trying to do it with these really massive phylogenetic trees that included tens of thousands of sequences, which we had available to us rather than maybe hundreds or thousands that we would typically use in a study. So we had information like the both the genetic surveillance as well as the actual incidence. We were able to build a really large phylogenetic tree and then classify branches as based on our region of interest, whether a branch was imported, whether it was circulating locally amongst within a region, or whether it was being exported from that region. That allows us to say, hey, uh, what what region itself is important as we're investigating an outbreak. So we used the state of Texas as a case study in this section to see how well our methodology would perform. We had this massive phylogenetic tree, and we were able to identify groups of viruses that were circulating locally within Texas, and the number of introductions within this region, as well as when it's circulating within a region, is it exporting out to the larger global population, or whether it's dying off there. When we did that, we were able to actually start looking at the transmission patterns within the state of Texas. These are rural urban classification codes RUCC1s are large urban centers, greater than 1 million population, and then RUCC2 and 3 are smaller, excuse me, are smaller populations, and then rural being the smallest. This classification reflects things like population density and connectedness of populations. And within Texas, the four largest urban centers, this being Houston, Dallas, Austin, and San Antonio, were the major source for outbreaks in the rest of the states. Okay, so if you can control the outbreaks within these larger regions, you can control the outbreaks across the entire state. That is, I think that is actionable information, and that should be used to determine how we are going to utilize resources. And this is information that we're able to extract directly from that phylogenetic tree. If we want to dig even further into the study or into these processes using some of those same metrics that we had, we want to identify individual clusters within a population, in this case being Harris County, so one county within Texas, and see how that county is related to other neighboring counties, as well as what's determining transmission within that county. We looked at age groups and gender, and we saw that age group was a major predictor for the spread, and that in particular, middle-aged adults were the primary source for all other groups. We did have transmission between these groups, but in fact, middle-aged adults were really important in spreading that virus to other groups in that population. Uh, when we looked at how that particular age structure was whether it was significant on the phylogenetic tree, it was. It was. So this is the association test that we developed for this study, and it was highly significant here. So it gives us some insight into how that virus was spreading within that single group, or within that single population of Harris County. So I'm going to end there. I want to highlight Luke Liu, who did a lot of the work to develop those cross-scale phylogenetic modeling. And of course, Ludi, who's been doing a lot of the coordination here. But we do have a number of other study areas that I'm just going to go through very quickly here. So Cody has been, he's a recent graduate. Both Luke, Cody, as well as Gabriella, who's right here, just graduated this year. But Cody focused on how do we integrate human mobility. Gabby has been looking at integrating wastewater into clinical surveillance. Sachin over here is looking at uh, avian influenza emergence in Asia. Tosin is a mathematician. He's looking at economic impacts of these emerging pathogens. Mohammed is doing a lot of risk mapping. He's another mathematician within our group, doing a lot of risk mapping of emerging pathogens, and hopefully we'll be able to use that for predicting where epidemics start, for example, influenza. Guppy has been focused on trying to make good decisions about building a data set. And then we have Christina here, who's looking at host factors and trying to provide the base information that'll be able to integrate it into these biomedical epidemiological transition methods as well. Okay, so the whole effort to try and develop this is very very big and complicated, but we're doing it in a piecemeal way with all these individuals. This didn't capture everybody, but we couldn't fit everybody in the picture. So I do thank you and some of the funding that we've had with these projects.